Well, good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Post-ASCO 2026, Translating Insights into Trial Development and Execution Strategies. My name is Ryan Muse, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank Worldwide Clinical Trials who developed the content for this presentation. Worldwide features a dedicated oncology business unit within their full-service global contract research organization. They partner with biopharmaceutical sponsors to deliver expert-led, globally scalable oncology trials across every phase of development. They help sponsors move fast with confidence and scale without disruption. Now, I'd like to introduce our speakers for today's event. Dr. Andrew Zupnik has more than 25 years of experience focused exclusively on oncology. As Vice President of Oncology Drug Development for Worldwide, he supports study optimization, delivery oversight, training, and the development of new initiatives across commercial and operational teams to ensure Worldwide remains at the forefront of industry trends and emerging oncology therapies. Dr. Heidi Gillenwater is a medical oncologist with a unique blend of 10 years of clinical and 18 years of industry experience in oncology drug development. Her perspective and contributions led to approvals and label expansions for Kyprolis and Breyanzi. Dr. Gillenwater has a proven track record of leadership, having held key positions at BioNTech, Lyell Immunopharma, and Juno BMS. Dr. Elise Horvath Walsh is a Vice President of Clinical Development at Kumquat Bioscience and a board certified hematologist oncologist with over 20 years of experience in medical hematology oncology, including 14 years solely dedicated to clinical research. At Kumquat, she is the medical lead for 3 novel products and multiple trials. During her time at Karyopharm, she was the medical lead for selinexor and in uterine cancer. Dr. Victor Moreno is the Director of Clinical Research at START Madrid FJD. He obtained his medical degree from the University of Complutense of Madrid in 2004 and completed his specialized formation in medical oncology at Hospital Universitario La Paz. He then obtained his master's degree in molecular oncology from the CNIO and obtained his PhD with European mention and highest commendation in 2013 for his translational research in rectal cancer. Now, before we hear from our speakers, we'd first like to hear from you, our audience, and we have a poll question appearing on screen right now that you can interact with. In order to participate, you can select one of the answers and then click Submit to register that answer. Right now, we'd like to ask you, which side of the industry are you joining us from? Your answer options are academia or government, sponsor, biotech, sponsor, large pharma, or service provider or investment. In a moment, I'll hand it over to our speakers for their discussion with you today. But for now, again, we'd just like to hear from you on the question of which side of the industry are you joining us from? I see answers coming in, and I'm going to leave it open a little bit for in order to get as many answers from you as possible. And we'll have another poll question after this, but we'll just leave this one open a little bit longer. I appreciate you all taking the time to participate in this. Looks like we have most of our answers, so thank you very much. Let's go ahead and look at where the votes have come down. It looks like we have a 9% choosing for academia or government, the 9% of you for sponsor biotech, and then sponsor large pharma, and then 73% for service provider. Very interesting. Thank you very much for your participation in that. And now our next poll appearing on your screen right now is the question of which theme coming out of ASCO are you most interested in hearing about today? Your answer options are targeted therapies, immuno-oncology, development in or from China, antibody drug conjugates, or trial design and patient recruitment. I appreciate you're all much faster at answering this now after that first question. So thank you for participating. And then I'll hand it over to our speakers shortly. But again, we just want to hear from you. Which theme coming out of ASCO are you most interested in hearing about today? Our speakers will be, of course, touching on the various themes coming out of ASCO, but very shortly. But for now, I'm going to leave this open a little bit longer, try to get as many of you as we can to participate in this. And we like to see these answers coming in. Thank you very much. All right. It looks like we've got most of the answers in. Thank you, everyone, for participating. Let's look at the results for this one here. It looks like we have 14% of you choosing targeted therapies, and then 14% also for immuno-oncology, 21% for antibody drug conjugates, and 50% of you choosing trial design and patient recruitment. Thank you again very much for your participation in this, everyone. Without further ado, though, I'd like to go ahead and hand the mic over to our moderator for today's event, Dr. Andrew Zupnik. You may begin when you're ready.
Perfect. Thank you, Ryan. Can you hear me okay?
We can
We're ready
indeed.
to rock. Thank you, and thanks everybody for joining us today. I appreciate the input on those polls. Those kind of help us shade the plan and some of the conversation for today. A little bit purposely designed because we're actually going to touch on most of those topics that were on the last poll there. But nice to hear that there's a large interest in the patient design, or study design and patient recruitment, as well as some of the targeted therapies and IO coming out of ASCO. Surprising, there were no votes for the China topic, which seems to be a hot one amongst this team. We're always talking about it. So appreciate everyone for joining. Good afternoon, good morning, good evening from where you're joining us around the world. I'm really excited to be here today, and especially to be joined by this fantastic panel. We have some brilliant folks who have a wealth of experience in the oncology drug development space. And we're here to talk about ASCO as well as some of the themes and trends coming out of that meeting. It's hard to believe it's only been 2 weeks, guys, since we're back from Chicago for those who were there. So whether you're on the ground and still recovering like myself, or watching from afar, there was a lot of data at this conference. I think it did not disappoint, much like some recent years. We had jam-packed sessions, compelling information, some standing room only crowds, and I think I'm still waiting on the final numbers. But I heard that they're tracking over 40,000 attendees for folks who were there. It's always Chicago. I think the nice weather this year was particularly compelling for how nice it was for that weekend. Nearly 3,000 posters, right? And over 450 oral presentations. So that's a lot, right? Tons of information. Obviously, there was a lot of themes and novel data coming out of that. And what we're here today to do today is to help summarize some of the key themes that came out of ASCO, as well as help translate the so what of that into where we think that sets trends for the industry, what we're planning to see around the future state of the industry, and how to apply that to those of us who are working in the drug development space, whether you're on the sponsor or the service provider side. So, with that, I'm really excited about the panel that we have here. A wealth of oncology knowledge on both the sponsor and the service provider, as well as the site side. So, we've got some different perspectives for you today, as well as different perspectives around portions of the world. We've got the West Coast, East Coast, and Europe covered with our three fantastic panelists. So, let's dive right in and kick it off. I'll be walking through some of the key themes and trying to parse the conversation into different topics. And then we'll unpack some of the data around ASCO as well as some of the so what of those themes. All right, ready? Ready, guys? So I think the first piece that we obviously can't talk about ASCO and not mention is the fantastic data coming out around daraxonrasib and targeted therapies. That'll be our first kind of main topic is targeted therapies. And every year it feels like the mainstream media picks up one or two results from ASCO, and you start seeing these in mainstream news feeds. I think Wall Street Journal, New York Times had articles around ASCO here in the States. It's always funny when I hear from my parents sending me a newspaper clipping. Weren't you at this conference? Yes, yes, Mom, we were there. But I think the biggest news was what we heard. I saw some news articles that said, using terms like unprecedented, a grand slam, a game changer for pancreatic cancer. So perhaps let's kick off around that topic. Elise, I know this is near and dear to your heart, having been in the targeted therapy space for a while. So maybe you can share a bit of the history around RAS targeting and the theme around drugging the undruggable was a phrase that we heard a lot around ASCO and some of the key results that came out of that main plenary session around pancreatic cancer.
Yeah,
Over
happy
to you,
to.
Angie.
And thank you, Andrew. Thank you for the introduction. Thanks everyone for attending. So, RAS actually is a fascinating molecule. So it's part of downstream signaling from tyrosine kinase receptors. And it's what controls when a tyrosine kinase receptor is activated, controls the downstream signaling below it, and therefore can, when it's mutated, lead to carcinogenesis. So RAS was actually, so obviously being where it is, it's a key driver mutation. It was first discovered back in 1982, where there's three different RAS molecules and one was discovered, the other subsequent. But it took until 2021 where our competitor, I was at Mirati with adagrasib, but the competitor was sotorasib, um, actually got approval. So it was almost 40 years that it took from discovering this essentially number one driver mutation to actually being able to drug it. And the reason it took so long is that RAS is a uniquely difficult molecule to target. So, unlike targeting our first targeted therapy was estrogen receptors. Very easy to target right on the cell surface. But RAS is a molecule that when it's mutated, it folds up. And so trying to get to the target, you have to design a drug that can get into this deep pocket. So it took a long time for people who are in drug discovery, the chemists, the brilliant chemists who actually create these molecules, to actually figure out how to target that. Um, and so, people have been waiting a very long time. Many, many millions of patients have been waiting a long time who have had these mutations and gone without really any effective therapy. They've received chemotherapy, checkpoint inhibitors, all kinds of indirect therapies, but targeted therapies means you directly hit the target and then you have a better response as a result and better clinical outcomes. And so sotorasib got approved in 2021, and then adagrasib got approved the next year in 2022. And so took us essentially 40 years. And so it just was a long journey that finally got fulfilled and was very, very meaningful. The approvals for sotorasib and adagrasib were first in non-small cell lung and then subsequently in colorectal cancer. And so I think that Andrew's probably going to want to ask some subsequent questions about this drug and its approval that will be impending, and its results. But it took us a long time to get here, and I think that the patients are grateful, the physicians are grateful, to the scientists who and the drug discovery units that finally got us here.
Yeah, do you want to highlight for those who maybe weren't at ASCO what the sort of key takeaways from the pancreatic results were? I mean, we saw a nearly
Yeah.
doubling of survival, which is what
Yes.
really stood out to everybody.
Yeah, and so for daraxonrasib, so pancreatic cancer, as I think most people know, it's a cancer with really dismal outcomes, and the median survivals have been, 6 months. Is an approval of Tarceva was only improvement of basically 1 month in survival and got an approval, but obviously that wasn't much. And so daraxonrasib, its response rates compared to chemo. So the trial was designed, it was very straightforward design of standard of care, meaning chemotherapy, and it was a dealer's choice of chemotherapy versus daraxonrasib, which is it's a RAS inhibitor. And it inhibits the on position, and then I can explain that a little bit. So RAS is a molecule that, based on its phosphorylation, is either on or off for the downstream signaling, and so it inhibits it in the on position. And the results are that the overall survival actually was doubled with the use of the RAS inhibitor compared to chemotherapy. And so in pancreatic cancer, that's pretty remarkable. considering that the prior approvals have been for far less and been obviously far less of an improvement. The other thing that's unique to daraxonrasib is that it is not a specific inhibitor. So soto and adagrasib are specific to only inhibiting, because the RAS mutations are point mutations, so single exon shifts. And daraxonrasib is actually what's called a pan-RAS inhibitor, meaning that any mutation basically inhibits. So, because pancreatic cancer, the most common is G12D, and actually in this trial, about 80% of the patients had G12D mutations, and then after that G12C and so forth. So this molecule can actually inhibit any of these mutations, being a pan-RAS inhibitor. And so in the large variety of mutations that we're seeing, it did improve survival by doubling, which was pretty remarkable.
Yeah, thank you. Maybe Heidi, I can pass it to you. You were there, right? I remember you sharing some of the feeling and the response from the crowd. How did that go?
Yeah, no, absolutely. I've been, as you probably saw from my intro, attending ASCO for quite a long time now, over 25 years. And I can count on my hands the number of times I've been in an audience where we've seen a response like this. It's very rare. I think in general, we're among, as oncologists, we're very optimistic folks, and we get excited about these in general, just small advances over time where we see months improvement in overall survival. So to see something like this, where we're doubling survival, where we're seeing a survival curve posted that shows at any point your chance of death is reduced by 60% based on this new therapy, it's amazing. And there was a standing ovation in this hall, where I think the majority of folks attending ASCO were. So it was really impressive. In fact, I think the speaker was a little worried he was going to go over his allotted time based on our continued standing and applauding at the survival curve he was presenting. But having said all of that, I think these trials where we see incremental improvement in survival or incremental improvement in response rate, they're all necessary. These early trials that you've mentioned, at least, where we're seeing improvement, but it's very modest. We had to do that work in order to get this home run. And so I think we all need to continue to be persistent and do the work, so that ultimately we get the home runs. And hopefully, with time, as we understand both the molecular biology and the chemistry of the drugs better, we will hopefully see these home runs more frequently. But yeah, no, it was an amazing experience to be a part of that. And also, Andrew, just as you mentioned, I'm the only oncologist in my family, but the number of family members who reached out to me because they had conversations with others hearing about these results, I think is a testament to the importance of this and where it's reaching, just mainstream audiences.
Yes, fantastic. Yeah, a significant impact. I think I'm seeing that this is already kind of opening the floodgates, so to say, around this concept of drugging the undruggable, or finally being able to go after some targeted therapies that hit some very fundamental pathways in oncogenesis. Maybe Elise, what do you kind of see around next steps here? Kind of what are next generation inhibitors, or where's the industry moving as a result of this pretty significant shift?
Yeah, so I think that there are still obviously other pan-RAS inhibitors that are being developed. There's also still specific, kind of like sotorasib and adagrasib, point mutation inhibitors that are being developed. So still some additional G12C and D. Also other point mutations are in development. There's also some interesting other ways of going about drugging RAS. So, as I mentioned, there's the on and off configuration. You can either block RAS and it's while it's in its on state, so downstream signaling, or its off state, or you can block both. So there's some of the next generation inhibitors are blocking both the on and the off state, so getting for more complete blockade. There's also increasing the potency, so you increase the cell kill preclinically, that hopefully translates into increasing cell kill clinically. And then making your drug more selective, so it has less off-target effects, so it has less side effects. So it's not hitting so many other targets. There's also some new RAS degraders that are being developed. So rather than targeting the mutant protein, it actually and just inhibiting it, it actually degrades the mutant protein and dissolves it. So there's a whole host of next gen compounds that are gonna come out as the version 2.0s and beyond for RAS subsequently. And then also, the biggest problem that we have with oncology in general, and RAS is not unique, but is obviously resistance to therapy. So, I think a lot of work that needs to be done in the RAS pathway is the resistance mechanisms. And there's a host of ways you can do that. You can also blockade the upstream, and you can, so the tyrosine kinase receptor in combination with the RAS molecule, or you can block further downstream, because a lot of the resistance happens from crosstalk. So below RAS, there's a whole bunch of molecules that then are turned on, and then they stay turned on, unfortunately, and they go back up. And so if you block those in addition, then you might actually get better clinical outcomes by getting that additional blockade. And then so I call it up, down, and sideways. And by sideways, I mean also doing other things that inhibit carcinogenesis. So of course, things like chemotherapy, checkpoint inhibitors, also cell cycle agents. There's a host of other approaches that you can take to try and either prevent resistance or treat resistance once it's developed.
I expect we'll be seeing a lot of novel approaches coming out pretty quickly. I'm seeing the enthusiasm around some of these new targeted therapies is high, which I think translates to some of our viewers. Some unprecedented enrollment rates are potential. Trials might be moving a lot quicker because patients are looking for and interested in these types of therapies. So maybe Victor, I can toss the ball over to you as actively practicing, kind of your take on the outputs from some of these results. Specifically the daraxonrasib. Is this something that you're even seeing people asking about? Is this something you're going to be offering patients as soon as it's available over in Europe?
Yeah, it's amazing. When we came back from ASCO this year, how many patients and families were asking for this, because the results were impressive. And it's true that we have a lack of clinical trials. We have many drugs in development, many KRAS inhibitors, but the slot availability in early phase clinical trials is not always it's very difficult, because we have a lot of clinical trial sites participating in these studies, and sometimes it's hard, and it will take some time for these drugs to be available in Europe. But I also like to chime in something that after ASCO maybe is also interesting for our audience, because it was not presented at ASCO, but it was a press release last week about the combination of the PRMT5 inhibitor, the Tango drug 462, in combination with daraxonrasib, with 90% response rate in this setting, patients with KRAS mutation and MTAP deletion, and in second, third line pancreas cancer. So these results are being impressive, and I think we're really seeing that targeting different pathways, not only KRAS, but additional and molecular alterations. As we make these drugs safer and they're being able to be combined, we're seeing even better responses, of course. But we want to see these compounds in earlier lines, in first lines, because that's where you can help most of the patients.
Agreed. I think that's what it's nice to see the industry moving towards earlier lines of treatment and really moving the needle for patients versus, like you said, Heidi, kind of um, small incremental changes don't have a significant impact. And this is where we're starting to see some major changes to overall, not just survival, but quality of life. So maybe I can pivot to another one of our topics. I'm going to skip one, guys, and move over to the immuno-oncology, if that's okay. That's another big chunk that our audience was interested in hearing about. So that's a large sort of class of drugs, immuno-oncology in general. Maybe, Victor, I can just start with you. There are quite a number of IO type therapies and themes coming out of ASCO. If there anything that you really caught your eye and you think is most interesting around some novel IO therapies?
Well, one of the studies that was presented by Fiona in ASCO, and I was co-investigator, so I have to disclose that. It's with the gray wolf, the GRWD 5769, which is an ERAP1 inhibitor, because the mechanism of action is totally different, and we like to see that. We're used to see just monoclonal antibodies targeting the surface receptors of the T cells, and for 10 years we have not been seeing a lot of improvements beyond PD-1, CTLA-4, and LAG-3. So it looked like all the field was stuck there. And this compound is totally different. The mechanism of action is an inhibitor of a protein, ERAP1, that it's an enzyme that trims the proteins to load them onto the MHC class 1. So the way this drug works is that you change the repertoire of peptides that are being presented into the MHC class 1. And therefore, the chances of having a T lymphocyte to detect a novel peptide presented there increase. And in this phase 1 study, we treated patients that were actually progressed on prior PD-1 therapy, and across different tumor types, non-small cell lung cancer, urothelial, hepatocellular carcinoma, and even MSS colorectal cancer, which is a pretty difficult to treat cancer with immunotherapy. Response rate varied between 20-30%. So pretty significant advance for still early. Obviously, it's a phase 1 study, but I think that it was very promising data.
Yeah, agreed. There was a lot of early phase data presented that was significant. If I can plug one as well, that was kind of a favorite of mine, and being a scientist by training, really novel mechanisms of action really caught my eye around the in vivo CAR T data that was presented at ASCO. This was data that came from Colonia, which is now Eli Lilly. We kind of got a look behind the curtain for why Eli Lilly acquired this company for, I think, upwards of $7 billion with a B dollars. And this is very early data, so nothing on the scale of daraxonrasib and pancreatic cancer, but in vivo CAR T has the potential to be a complete game changer around mechanism of action as well as ease of delivery. And in a very small patient population, we saw 100% overall response rate. So 18 out of 18 patients had a response rate, and many of those were complete responses, as well as what was pretty novel with this was 100% were minimal residual disease after 1 month. So this is the promise of taking. We've seen CAR T and the potential benefits of it in hematologic malignancies for years, but as everyone knows, there's a lot of complexities around manufacturing. Patients have to go through apheresis, lymphodepletion, and there's now a new technology around basically using a virus to do this all within the cells, so you don't need any of those steps. The manufacturing components are removed, lymphodepletion and apheresis can be gone. It's basically a one-time gene therapy, and you're done. To see that sort of proof of concept data presented was, to me, really fascinating. And I think a space that we're seeing the industry is moving a lot of interest into what's called in vivo CAR T versus ex vivo, which is how we've been doing, autologous or allogeneic treatments to date. So that really caught my eye, and I think that's a new theme that we're going to see. We've already seen a lot of investment in companies that are in vivo CAR T focused technologies. And perhaps it's finding those solutions that then maybe are combinations like we're talking about. You've got an immuno-oncology approach, perhaps combined with the targeted therapy approach down the road, can be kind of the future state for the industry. But that's one trend that I'm seeing, and a lot of enthusiasm around this pretty unique space. Maybe we can pivot to another topic. Another favorite of mine is ADCs. That's been a hot topic at ASCO for the past 3, 4 years. Also, I think a few years ago, a standing ovation, right, around some of the early novel ADCs, antibody drug conjugates. And so what we're starting to see this time is kind of what I call the next generation of ADCs. We've got bispecific ADCs, we've got novel payloads, novel antigens, different linker technologies, and all of this is around kind of optimizing the therapeutic window of ADCs and finding the best possible fit. So maybe Victor, we can start with you. Um, One of the trends we saw at ASCO was taking the ADC data that we saw a few years ago and slowly moving that closer to first line therapy, which is where, like we said, can really make an impact for patients and significantly change the outcomes or the survival curve as well as the quality of life. What did you see there at ASCO around maybe breast cancer? Because that's where we've seen the most ADC news.
Yeah, they then presented the update results of the Ascent 04 with sacituzumab, govitecan plus pembro, first line, in comparison with chemotherapy plus pembro. And there was a significant increase in progression-free survival hazard ratio of 0.65, so very significant increase in the progression-free survival. And something that is always a question here is, okay, we're increasing PFS, but what happens after that? And the data they presented was interesting because they maintain this benefit in PFS too. So despite the patients being able to cross over, they show that receiving the ADCs before is better than chemotherapy. So that's very encouraging data for ADCs in the first line setting. There were other ADCs that are also very interesting, and I think they were less practice changing, but maybe also worth to mention, like isalantamab vedotin that was showing in second line of esophageal squamous cell carcinoma, an improvement also in PFS. This is an EGFR HER2 ADC, and the other bispecific TROP2 HER3 drug, the JSK and 016, that shows some good responses in breast cancer. Obviously, those are in later lines, but I think that we had a lot of, as you say, novel ADCs using this bispecific design. I think it's very interesting. Yeah.
Yeah, that seems to be the thing. I mean, there's a ton of interest in ADC development. Heidi, you're in the thick of that. Maybe some other novel approaches within ADC technology.
Yeah, sure. Yeah, no, I think it's interesting to look at the history of ADC development and back in the early days when Mylotarg was approved, and I think we consider that a targeted therapy. It's novel, linking an antibody to the target, bringing it to the tumor cell. But in reality, I think what we've learned is that there are certain aspects of the ADC that can be really important in making these truly targeted therapies. That meaning delivering the toxic chemotherapy to the tumor cell. I think if you look at the PK data for Mylotarg, you're seeing a lot of systemic exposure by the payload. So now I think the other trick is designing new molecules that will be effective in solid tumors. So I think part of that is understanding the potential targets and the complexity of the targets in solid tumors. So I think one theme is either looking at dual targeting or just novel targeting. One of the I thought interesting um, abstracts that was presented, it was a phase 1 study of an ABI 969 molecule that targets both PSMA and STEAP1. So these are two targets on prostate cancer that are very highly expressed. There is quite a lot of overlap, but there are some patients that have a dominance of one or the other target. So having a molecule that targets both, I think, is a really smart idea. They also presented very nice results. It was a phase 1 trial, so we're seeing the dose escalation portion of the study. They're moving into dose optimization. The interesting thing that they saw, that they reported, was that they did see dose-dependent toxicity, but fairly almost universal efficacy at all dose levels at or above 3.0 milligrams per kilogram. So quite a lot of efficacy, both in terms of PSA response as well as resist response. It was they called it a novel topo 1 payload. So we don't have a lot of details on the specifics of that, but of course, a lot of ADCs are using topo 1 payloads. And as you would expect with that payload, quite a lot of heme toxicity, particularly anemia in this patient population, prostate cancer patients who come into trial with quite a lot of anemia. So that's not surprising. At the higher doses, I would say unacceptable grade 3 neutropenia, so over 50%. But when you get down below their highest dose levels, I think they're getting into ranges that are acceptable. But their overall response rate in terms of both resist and PSMA or PSA responses are quite encouraging. So it'll be interesting to follow this molecule as it moves forward. As I mentioned, they reported that they're in dose optimization, and I suspect we'll see soon plans for further, more pivotal development for that molecule. The next to that, oh, sorry, go ahead.
No, there's another one. Go ahead.
No, you can, if you have a follow-up specific to that.
I was going to think, I mean, it seems there's a lot of toggling of the variables with ADCs, right? You've got the target, the target antigen, the linker, the payload. Do you feel like we're still going to see some pretty significant improvements, or are we kind of fine-tuning things right now because have we already identified the biggest game changers, or there's still some new ones out there?
I do think, if you look at AACR, the preclinical world is really so full of different, I think, very interesting and exciting options. So I don't think we're there. I think we're really at the tip of the iceberg on that. Victor, you're probably exposed to a lot more of the different trials that are out there than I am. I'm fairly narrowly focused on what we're doing, but I do think that we're still in that fine tuning before we get the home runs, if you will.
Perfect. I think there's a lot more to come. I get the fun job of seeing a lot of protocols.
You too, I should say. Yes, of course.
Pretty unique approaches to novel payloads, like you said. Not everybody has to do a topo 1 inhibitor, and unique approaches to masking perhaps more toxic payloads or dual targeting, or you can get crazy with your antibody designs. I've seen some quadra-specific antibody ADCs, and that's where you really have to try to balance that safety profile because you can impact quite a number of pathways, which is the intent. We can really move the needle on seeing some effects for patients. Did you have another one, Heidi, that was kind of a highlight from ASCO?
The other one that I thought was very interesting, I think some based on the target, but also the results. So a NaPi2b ADC that has an exatecan payload in ovarian cancer. So obviously a really tough patient population to treat. I would say they had a very nice safety profile. And this one, in contrast, had, if you go up into the higher doses, again, heme toxicity was their chief, most common toxicity, but they were able to see, again, efficacy at various dose levels. And I think they landed on a dose level of, I think it was 2.1 milligrams per kilogram as their dose level to bring forward to further development. But their activity was quite high. They had an overall response rate of 61%, and again, in a very, very difficult to treat patient population. So, the vast majority of patients at all, most dose levels had a response. So I think very encouraging.
Agreed. I think we're going to continue to see ADCs on the platform at most conferences for quite a while. All right, perfect. Maybe I can pivot to another one of our themes or topics. Victor, the audience was curious to hear about some of the impacts around, from a patient perspective. And I like where some of the themes we saw at ASCO, and certainly where the industry has been going, is around optimizing dose and perhaps the concept of doing more with less, right? So there were a couple of studies, maybe Victor, if you saw a few that were digging into that. Maybe you don't have to treat the patients with as much possible drug as you can, but it's fine tuning that optimization and what you're seeing from your perspective as a practicing clinician.
Yeah, since the Optimus project started to change all the protocols in dose escalation, we're seeing a lot of this, and it's very complex to how to fine tune the protocols. We saw a first wave of very complex protocols with huge expansion phases, those optimizations, those levels. And I think now the industry is realizing that maybe that is not necessary, that you can get a lot of information about the toxicity from the dose escalation portion of the trial. And with the current point designs that we are seeing in most of the dose escalation studies, uh, we are doing backfilling cohorts. And out of those backfilling cohorts, you can get a number of patients that it's well enough to make sure whether that dose level is safe, it has the PK and PD data that you really need. And you probably don't need to randomize, 60 patients at two dose levels to be confident about what is going to be your recommended phase 2 dose. So that is the way I see that currently the early phase clinical trials are developing in terms of dose design. Something different now is what we saw there in ASCO. There was phase 3, very large phase 3 trials about de-escalation of therapy. So totally different topic that may be worth to highlight, like the Optima trial, the phase 3 with the in HER2 negative, ER positive HER2 negative breast cancer, where using the Prosigna, the PAM50 test, patients could avoid chemotherapy. And that is something that we also need to know, not only to optimize the dose of the drugs, but sometimes even to avoid therapies at all. So that's totally another topic that maybe thanks now to the ctDNA, this type of other tests, we can improve our management for patients.
There was also, I think in bladder cancer, another similar one, Victor, around kind of preserving patients. I like the theme of do you even need to treat, or can you do less and still have the same level of outcomes?
Yeah, well, the organ sparing, that was with durvalumab to avoid the cystectomy, and that is obviously a very very important studies, because they improve significantly the quality of life of patients. So trying to do less, well, it's not less, but even if you avoid such a surgery, is definitely an improvement. And as we improve our systemic therapies, I think that's where we have to go. And we will be seeing this type of studies more in the coming ASCOs.
Perfect. All right, I'm throwing you guys curveballs here. So another topic that the audience was showed interested in hearing was kind of patient identification, enrollment. Victor, you're closest to this, seeing this every day. What are the impacts of some of these themes that you're hearing from your perspective, being at a site that's also part of a growing site network? All of these new therapies that are coming out, like you said, are you getting patients asking for these treatments? How can how can we think about patient identification and recruitment to speed up things even further in oncology development?
Yeah, well, it might sound strange for some of this audience to hear this, but we need that all patients have the next generation sequencing as soon as possible. This is still in the United States, that's something that is very common, but in other parts of the world, not all patients have accessible NGS, and that's in tissue. We don't even think about cell-free DNA doing liquid biopsies. And this is critical to identify early on patients that are candidates for targeted therapies, it makes no sense to put them into a phase 1 study after 7 lines. They're not going to respond, or the tumors have acquired other mutations. So that test that the patients did on the primary tumor is not even of any use at that point. And that's about sequencing. But now we're talking about ADCs, and ADCs sometimes require immunohistochemistry. They require to do a new biopsy to measure if the target is there. So the tissue is becoming an increased problem in the management of patients to be involved early into clinical trials, and that is critical because so we reach out to our referring physicians to let them know that they should refer them as early as possible, because if we need to do this kind of test, sequencing, immunohistochemistry, we cannot wait until the patient is in progression, and then patient come and we say no, we have to do a biopsy, we have to wait 3 weeks. Patients will not be willing to enter the clinical trials at that stage. So they need to be aware of the studies as soon as possible.
Absolutely. Elise, Heidi, anything you want to add from your perspective as clinicians?
Yeah, no, I can reflect back onto a clinical trial that I was running that required a biomarker test, and I can confirm that the biggest roadblock was it was a biomarker that was not routinely tested for. And so, and it took, the turnaround time was, a minimum of 1 week, but getting tissue to the lab, that was another, it seems, 2 or 3 weeks. Um, Patients with end stage 4 lung, stage 4 cancer in general, they can't wait that long. And so we ultimately developed a standalone screening study so that we could get our pool of patients that had the biomarker. And unfortunately, this biomarker was somewhere between 25 and 60%, depending on the tumor type. So we screened thousands of patients in order to find those few patients that were at least potentially eligible based on their biomarker. And then, of course, they need to fulfill the rest of the eligibility requirements. But yes, this is it's an important process that we need to figure out and get more streamlined in order to efficiently identify the right patients for the right treatments.
And I mean, to follow on Heidi's remark, I think the word efficient. So, I I started with a great experience that then I never saw replicated, but it's your relationship with the companion diagnostic and the lab that does it. So in CLGB 10603, which is the ratified trial, we actually, it was an academic trial, it was an NCI trial, and so our lab was actually Ohio State. And so we actually turned around FLT3 testing within 48 hours, because these were acute leukemia patients. They cannot wait. As all clinicians know, these patients are extremely sick. They have to start treatment right away. And so there was a commitment between the lab and the sponsor, being the NCI, that we would get the testing done within 48 hours for all those patients. And we screened and enrolled 700 patients, and that those tests would be done in 48 hours. That makes it doable. Nowadays, biomarker testing is traditionally, a lot of it's done by commercial labs now, and that's where unfortunately you see the slowdown. And so if sponsors can partner early with the labs that are doing their biomarkers, or whatever tests are relevant to getting the patient the treatment, so that there can be rapidity, then you can get patients into trials quickly with central testing. But the thing that a lot of sponsors are doing now is and I've worked with this myself, is accepting local testing. And then what you do is what's called bridging later. And so you accept the local testing result based on it being done in with under acceptable circumstances, and knowing with 95% certainty that yes, that is correct. And then you do what's called a bridging study later, where you do all of it retrospectively. You do the central testing. and that really allows the patients to get on quickly because it allows whatever testing is done in a variety of settings, be they academic, be they non-academic, be they US, be they ex-US, it doesn't matter, and get the patients on quickly, and then do the central testing later. And then 95% of the patients were truly eligible for the test. And so it's just develop, opening your mind up to the best way to get this done as quickly as possible for the patients because they just can't wait.
Agreed. I love that it's all with the patient in mind and thinking about how to get data around the genomic, status of those patients and get them targeted therapies, as we're seeing the industry's continuing to move towards what targeted or personalized medicine, and that's where we're really moving the needle. All right, I want to be mindful of the clock and make sure we have a bit of time to get to some Q&A. I have one last question I like to ask everybody. We can't have a session around kind of highlights of or future state for the industry without kind of putting a bit of the crystal ball out there. And so I'll just go around the room here. Maybe, Victor, we can start with you. If you had to pick one key topic or something that you would think would be a plenary session or that you would love to see as a highlight of ASCO 2027 next year, what would be something that you would sort of forecast or predict will be a key topic a year from now?
I'd like to see cell therapy in solid tumors being a plenary at ASCO. I think that's the next frontier we need to cross, and we are seeing some of the advances like you mentioned before with the in vivo CAR T. I'd like to see that benefit that we see in the hematological world being reflected in solid tumors. And we're seeing some with T-cell engagers, CD3 bispecifics. We're seeing something. So if cell therapy is not in the plenary, I hope next year we have a T-cell engager in solid tumors.
Agreed. Maybe Elise, over to you. What are you kind of predicting or would love to see next year?
Oh, well, honestly, of course. I mean, selfish to say, I'd still love to see further RAS development. It's just been a molecule that I've been very passionate about for a while, and so would love to see that occurring. And again, being just a mutation that affects so many patients, and then it reaches so many. But there's just a variety of, and just honestly, whatever can impact the highest number of patients.
Agreed completely. I'm a little selfish in that boat of being a P53 researcher. Now that we've figured out how to drug some of these undruggable targets, I'd love to see MYC or P53 on the spotlight one of these days. It's been 70 years since we've
Yeah,
tried to find a therapy.
yeah.
Why to see P53 activator? That was in already. I think it was in plenary.
Yeah, we're getting close. Heidi, how about you? What's your crystal ball say for next year?
Oh boy, for next year, just on the horizon, I would love to, of course, being very biased now, love to see ADCs, can't speak anymore. ADCs reach the plenary podium, selfishly. Yeah, no, I think there's a lot of really interesting, I can't tell you which one specifically, but I think a lot of interesting development in that space. This is very generic, but every conversation I'm having these days, AI is mentioned. There's many aspects where AI is being involved in our lives now. But I think hopefully AI will help us identify the molecules or identify the specific conformation of the molecules that are most appropriate for future drugs. And maybe that will speed our development. And maybe we have some result based on this. Yeah, I think there's a lot of potential.
Agreed. AI will absolutely be on the list. Maybe Ryan, we can bring it back to you and make sure we hit some of the questions from the audience. I bet AI is in there somewhere anyway. I'll
Yes,
pass it
well,
over to you, Brian.
Yes, thank you so much, all of you, for this very insightful presentation today. As we move into the Q&A session, I'd like to direct the audience's attention towards the handouts module, where you'll find some additional documentation related to today's presentation to download for some further reading. I'd also like to invite the audience to keep sending their questions or comments right now for the Q&A session of the webinar. For those on GoToWebinar, use the questions window, and those watching on LinkedIn Live events, feel free to post your questions in the comments section. Now, I have already received some questions from the audience, so we'll get ourselves started with those. And the very first question I have for you here says, one of the topics at ASCO was the NHS Galleri ctDNA study. The question is thoughts on that and where or when early cancer detection may have a place in mainstream care?
Maybe Heidi, I can turf that to you, but I think this is one I was watching, and I think I would love to see us finally move the needle on making early cancer detection is the one thing that can really significantly impact outcomes for patients. So this was a very large trial. I don't think it read out entirely positively, though. So any thoughts on how that played out?
Yeah, no, agree. Very interesting. I think we would all of us would love to see number one, cancers being diagnosed at an earlier, more treatable, curable stage. And that's where, typical screening, breast cancer, et cetera, has made a difference. And just to outline for those of you who may not have attended that session, this was a huge trial. They randomized over 140,000 patients, so over 71,000 patients in each arm. And that was the observation arm versus the intervention arm. So the patients who are randomized, and these are patients that are healthy, no sort of particular risk factor for cancer in general, but the intervention arm had they had blood work, screening blood work on a yearly basis. And if they had markers that were detected, they were unblinded and sent for evaluation. They were not, the evaluation for the cancer was not dictated, but they were referred to a cancer center for screening for that particular cancer, which had been identified as a potential based on the blood test. Their primary endpoint was reduction in diagnosis of stage 3 and 4 cancers. And just by way of just overview, I think we all know that the bar for a screening test to be acceptable is high, and it must be a test that results in reduction in mortality. And so the rationale for choosing stage 3 and 4 cancer reduction diagnosis was basically a surrogate for reducing mortality over time. But they wanted to do this study in an efficient manner. So these results were read out basically, again, 3 annual tests, so read out fairly quickly with a plan to follow these patients over a longer period of time. So unfortunately, this did not reach its primary endpoint, meaning reduction in the number of stage 3 and 4 cancers diagnosed. But there were some interesting findings. Over time, there was an increased number of patients with early stage cancer diagnosed. There was a reduction in stage 4 cancers diagnosed. So those kinds of things make us all feel good. We would all like to diagnose fewer stage 4 cancers and more early stage cancers where we can quickly treat and hopefully cure those patients. The gold standard being, have we reduced overall mortality? We don't know that answer yet. This will be followed over time, but I think it's very interesting and really deserves follow up. And I think as we perfect the testing as well, there's just this is a huge area of investigation with different technologies, and we're getting better and more sensitive and more specific tests along the way. So I think, something to follow. Sorry for being so long-winded.
Really, really curious to see that data set evolve, because I think that's where the solution is in helping catch on cancer early. Ryan, I think we're out of time, so over to you.
Yes, thank you very much for that very insightful answer to that question. We are at the end of our time today. Note to our audience that if we couldn't attend to your questions, the team at Worldwide Clinical Trials will follow up with you. Or if you have some further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you click the exit button, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com so that you can register for this event and then gain access to features such as viewing the recording and more. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.